Thanks. Yeah.
Good afternoon. Thank you, Rick. There actually are some seats way up in the front row. The speaker will not bite if you're looking for a place to sit, and it's got lots of leg room. First, so I remember, I'd like to say again that today's lecture, as yesterday's lecture was, is made possible in large part because of the generosity of the storers, Ruth and Tracy Storer, who established an endowment that allows us to bring in leaders in the biological sciences. And Scott Edwards certainly is a leader in the biological sciences. I'll make this very short because I'm sure you'd much rather hear Scott than me. Scott got his undergraduate degree at Harvard, his PhD at Berkeley with Alan Wilson, then after some other positions is now back at Harvard, where he is in the director of the MCZ. And as Rick Grossberg was reminding me, not only is Scott a member of the National Academy of Sciences, but he already has a student who's a member of the National Academy of Sciences. And he's also a member of sort of every other society you could think of. He's been the president of 3 different scientific societies, so he's not only a great scholar, he's a great citizen, and served for 2 years as the head of the biological infrastructure at the National Science Foundation. And after his truly wonderful talk yesterday, which was aimed at a bit more of a general audience, today he gets to delve a little bit deeper, and we're really looking forward to your talk today.
Thanks. Thank you all for coming again, suffering through another hour of birds and DNA. I'm really excited to be here. I've met a lot of new folks and seen a lot of old friends, so thanks all of you. It's been a wonderful visit. Today I'd like to tell you about a recent project we've been focusing on, looking at the evolution of flightlessness. But I first want to introduce you to the cast of characters that have sort of kept me going these last few years. I think, as many of you know, I have sort of wide interests in evolutionary biology, mostly of birds, but sometimes their scaly, reptile-y ancestors. I sort of cut my teeth in evolutionary biology during my PhD working in Australia on phylogeographic questions. And more recently, we have a number of projects working at different levels in biology. One is perhaps in the category of ecological genomics, focusing on the evolution of resistance to pathogens. And we're studying a system wherein house finches, a small songbird, native here in the western US and introduced in the east, has experienced an epizootic by a bacterium called mycoplasma. So we're studying how the bird has evolved in response to this novel pathogen, doing quite a bit of work with gene expression and sort of whole genome scans, looking for evidence of natural selection. The work I'll tell you about today sort of falls into the latter 2 categories up there, involving comparative genomics and phylogenetic methods. And it's been a really fabulous journey. I've been fortunate to work with some very talented folks, some of whom have flown the coop. The lab this is sort of a previous generation photo, but Tim Sackton here, well known I think to many of you, got his PhD at Cornell, and he was a postdoc for a bit working on the project I'll tell you about, and then segued to a research scientist position in the computing core at Harvard. And he's really driven the majority of the analytical results that you'll see today. Phil Grayson is a 4th-year graduate student, and he is really taken over the empirical side of the project, not only sort of setting up the genome sequencing experiments, but delving deeply into sort of evo-devo studies, looking at developing embryos of chickens and ostriches and rheas, and I'll tell you about that stuff today as well. We've great group. Simon Sin is a postdoc working on genetics of storm petrels, actually in collaboration here with Gabby Nevitt. And Allison Schultz is the one for doing the house finch project that I won't talk about today, but which is quite interesting. Both Cassie Stoddard and Christy Riel were so-called junior fellows, so these Uber postdocs. In the case of Christy, she actually didn't have a faculty host when she came to Harvard, and she was interested in birds, and so needless to say, I co-opted her and brought her into our group. They are very fortunate both to be at Princeton now, working on questions of behavioral ecology and sensory biology. So it's been a great group, and what I want to tell you about today is sort of one of the bigger projects in the lab, looking at the evolution of flightlessness in this very charismatic group of birds. Forgive the acronym at the top. Those of you who were at my talk yesterday will know, of course, what CNEEs stand for, but we'll go through that detail in just a bit. The ratites and the Palaeognathous are a really iconic group of birds. As you saw in that big tree that I put up yesterday on the screen, they comprise the deepest branch of living birds. And so you've essentially got the Palaeognathous, which consists of all of these guys, including tinamous, which I'll tell you about in just a bit. The Palaeognathous is this entire group, which are essentially the sister group to all the remaining approximately 10,000 species of birds. So they're a very evolutionarily old group. They're also very enigmatic in the sense that, there are vastly fewer species than the sister group. We're talking about 13 species of extant ratites. Those are the flightless species, along with about 50 species of tinamous, which are volant. They are able to fly. But imagine that group of about 60 or 70 species being the sister group to all the other 10,000 species. So clearly you've had different rates of diversification and extinction in these two groups. And in many ways, if you want to call yourself a card-carrying ornithologist, you got to work on this group at some point in your career. They've tripped up many a big name in evolutionary biology. Darwin thought a lot about them. Thomas Henry Huxley kind of brought them to the fore, to the attention of biologists. And even in my graduate days, Alan Wilson's lab at UC Berkeley did a lot of really important phylogenetic analyses of these groups. But it's only in the past really 10 years that we've come to grip with their phylogeny on the one hand and how we can exploit that phylogeny to look at the issues of phenotypic evolution. Now, flightlessness is a composite phenotype. It's registered by many different aspects of the skeleton, body size, even the behavior and feather morphology of these species. Some of the kinds of things that we think of when we think of flightlessness include modification of the forelimbs. And so, for example, there have been a variety of studies looking at digit loss in different groups of ratites. When I say ratites, I'm referring to the flightless Palaeognathous, which also includes the tinamous as well. And there's actually a wide range of the extent to which forelimb elements have been lost. So, for example, the extinct moas of New Zealand, which we know now based on morphology and molecular data are clearly Palaeognathous birds. They're actually the only lineage in which the entirety of the forelimb elements have been lost. But of course, as many of you know, things like ostriches and emus, ostriches use their forewings for display, and so they have, quite a bit more skeletal elements in those forelimbs than other groups. Another key skeletal consequence of flightlessness is the loss of this keel. So as many of you are aware, most extant birds will have this very dramatic keel on the sternum. And of course, this is thought to be a prerequisite for powered flight. As you saw in the talk yesterday, many ancestral theropods had breastplates, and even furcula, the clavicle, but no keel. And so the loss of this keel is very much a signature of flightlessness. Some flightless groups, such as penguins, I mean, penguins are technically flightless, even though if you view them underwater, it's clear they're flying in a heavier medium. Penguins have a very substantial keel. And so flightlessness doesn't always result in this, but it's clearly one of the most consistent features within the Palaeognathous birds. And then finally, you've got these vast changes in body size. So for example, the elephant bird of Madagascar, which has been the subject of a few recent phylogenetic studies, is clearly a Palaeognathous species and was essentially the largest bird to have survived into modern times. If you go to Madagascar, you can see striking evidence of its very recent extinction only 500 or so years ago, eggshells strewn all through the dunes and whatnot. And we know that these birds stood 10, 12 feet tall, remarkable examples of island gigantism. At the same time, you've got very diminutive species like kiwis in New Zealand, which are quite small, though displaying quite a range of body sizes. So there's a lot of phenotypes that sort of fall into this category of flightlessness. And I think that's one of the challenges of the project, but at the same time, it's one of the opportunities. Now, for the vast majority of Palaeognathous systematics was studied under the assumption that the flightless Palaeognathous, the ratites, were a monophyletic group. And so if you look back at the older literature, I mean, Joel Craycraft was, I think, important in sort of establishing the modern phylogenetic study of Palaeognathous. You will see scenarios wherein tinamous are placed as the outgroup relative to a monophyletic Palaeognathi, these flightless lineages. And in many cases, even in many molecular studies even, it was sort of a knee-jerk reaction to use the tinamous as an outgroup for the Palaeognathous so as to root the tree. And of course, that will enforce a situation in which ratites are monophyletic, and you can begin to think about scenarios of distribution via the spreading of the Gondwanan continents, which is sort of the dominant scenario for the biogeography of the ratites up until just a few years ago. Now, fast forward to 2008, when a very important study, sort of byproduct of one of the early phylogenomic studies of all birds, led by Shannon Hackett at the Field Museum, took the novel perspective of, well, let's not force tinamous to be the outgroup. Let's see where they fall in the sort of overview, using sort of successively deeper outgroups of using other groups of birds. And lo and behold, the striking and almost unbelievable conclusion, at least for ornithologists, was that ratites lost their monophyly. So essentially there were some ratites which were thought to be more closely related to tinamous than they were to other ratites. This may seem like a sort of a trivial shift, but as I'll show you, and I hope to convince you, it suggests strongly that loss of flight may not have occurred as a single event in the common ancestor of ratites, as sort of the Craycraft scenario would suggest, but rather likely occurred multiple times throughout the tree. So that initial study was 2008, and it's been recently corroborated by whole mitochondrial genome sequences, this by Alan Cooper's lab in South Australia. And as you can see here, we've got this interesting situation wherein tinamous appear to be sister group of moas. So remember, tinamous are these volant, chicken-sized birds, about 50 species, spread out all through the neotropics. Sister group to the moas, a group of about 11 or so species, entirely extinct now in New Zealand, driven to extinction within a few hundred years of the arrival of Maoris in New Zealand. That said, very spectacularly detailed fossil evidence, very high quality DNA information, and as I'll show you, we've been able to prepare a draft genome of one of the extinct moa species, the sample of which was probably about 2,000 or 3,000 years old. So then you've got your elephant bird, and you've got kiwis here, kiwis from New Zealand, elephant bird in Madagascar, and here you've got your emus and cassowaries, both from Australia, and then the rheas, and then the ostrich right at the bottom. So two things are important here. First of all, the biogeography makes absolutely no sense, and I'm not going to talk any more about it because I don't think we've figured it out. The dominant scenario now is one, really sort of the second point is that unless one's willing to propose that tinamous regained flight from an ancestor that perhaps lost flight right at the base here, one is forced to conclude that, in fact, flight was lost multiple times. And so Cooper's group, as well as several other groups recently, have proposed that flight was lost multiple times, not only in the extinct lineages, but in the extant lineages as well. So, and I think this would be a fun discussion. I mean, not everyone, in particular, Joel Craycraft, whom I talked to last week, not everyone's convinced of this scenario. Is flight one of those complex characters that we imagine is much more difficult to evolve than to lose? Well, personally, I think it is, and I think the weight of the evidence suggests multiple losses of flight. But at the same time, I haven't performed a formal test, and I probably haven't stated all my assumptions completely explicitly, which is something that Joel likes people to do. But I should say, though, that the motivation really and sort of the experimental design of this project is predicated on there being multiple losses of flight. We actually think that, and we are in a position to test the hypothesis by looking at the genome, that in fact flight might have been regained in tinamous. We haven't really done that yet, but I think it's entirely within our grasp. So to sort of prepare a genomic resource for this project, we embarked on a series of whole genome sequencing studies using standard Illumina sequencing approaches, as well as the all-paths assembly. For those of you who do genomics, you'll know what I'm talking about. And we've produced a set of 10 new Palaeognathous and tinamous genomes. So most of these samples came from within the US. It's very difficult to get samples from many of these species overseas. But in general, as I'll show you, the quality of the genomes is quite high. And it's provided a really useful foundation for these studies. We also were able to inherit a draft genome from a little bush moa, again, one of this extinct group from New Zealand, produced by Alan Baker at the Royal Ontario Museum, who was a collaborator with us from the start. Sadly, he passed away in 2014. And so, but it's a very good quality genome for an extinct species. It's about 5x coverage, and although we haven't incorporated it into our main phylogenetic analyses, well, we have it in our tree, but because of its quality, we haven't incorporated it into the whole genome alignment, which I'll tell you about in just a second. But it's provided a critical kind of extra node, I would say, for this analysis. And really the only group of Palaeognathous that we haven't looked at is the elephant bird from Madagascar. So just a little bit of sort of technical stuff. For those of you who, people say they sequence genomes willy-nilly. We're doing it. Everyone's doing it. But let's look at the quality of some of these genomes. And what you see here is one measure of genome quality, namely the Contig N50 size. And here you have on the x-axis a whole series of genomes produced mostly by the Avian Phylogenomics Project, which published their 50 genomes results that I talked about yesterday back in 2014. And what you can see is that the size of the contigs in our project, indicated here in red, are significantly high. They're robustly high. I think they score well with sort of contemporary genome projects. That said, of course, as we all know, technologies are changing, and we fully expect even these moderately good genomes to be surpassed in the near future. But I think for looking at the kinds of questions we're looking at in terms of generating phylogenomic markers and looking for genes and regulatory elements involved with flightlessness, it's a very adequate data set. And we're also applying these approaches to other groups. So one of the first things that Tim did was to set up a whole genome alignment of about 42 species of birds, 10 of which were from our work and about 32 of which were from the Avian Phylogenomics Project. And just a few details, whole genome alignment is a computationally very intensive exercise. In the Avian Phylogenomics work published in 2014, they used an approach which essentially was sort of a pairwise approach in which similarities between species were used to create subalignments, and then were all summarized in reference to a single reference genome, which was the chicken, which has the best sort of annotated genome of any bird. In Tim's case, we used a different protocol called progressive cactus, designed by David Hauser's group at UC San Diego, which essentially uses a phylogenetic scheme to not only align pairs of genomes along the tips of the tree, but essentially estimate ancestral genome sequences moving down the tree. And it's very advantageous because now we're not stuck to just a single reference. We can sort of study any species in any comparison we want. So what you can see here is the browser where we have various statistics up here. All of the blue regions are conserved relative to the reference that we've chosen for this display, whereas the red are point mutations. We can actually find lots of evidence for little inversions and whatnot. Here's an example of a gene, DMRT1, which is well known as being important in the sex determination pathway in birds. We can find many genes. The genomes are very complete, and so we're very pleased with this result. And as soon as this is released, hopefully within the next few months, it'll be a really important resource for evolutionary biologists working on birds. Here's some examples of some of the estimated ancestral genome sequences. And so we can essentially look down the tree and ask, well, what was the likely genome sequence of the common ancestor, say, of emus and cassowaries? And that's a really nice thing to be able to do. Now, the first thing we wanted to do was to establish the phylogenetic relationships of this group. And here's just a number of hypotheses that have been around. You just saw this one before. And it's really the rheas which are turning out to be the bugaboo in this group, the rheas from South America. They tend to jump around in different parts of the tree. And intriguingly, I should say, usually not with very strong support. There's something going on with either the rhea genome or the way the data has been analyzed, which seems to be problematic here. So we essentially subsampled this whole genome alignment, not just for the 10 ratite species, but essentially for the entirety of the 42 genomes that we looked at. We looked at a ton of markers, and so we looked at 3 classes of markers, one of which are these conserved non-coding elements, which I'll tell you about in just a second. We also looked at introns, those spaces between the exons and genes. And then finally, we looked at a class of marker called ultra-conserved elements. And for those of you that do phylogenetics, you'll know that ultra-conserved elements are a very popular class of marker that is abundant in vertebrate genomes and sort of easily retrievable by techniques like hybrid capture and stuff. Now, this is really a vast data set. It essentially covers 3% of the entire genome. And, I think the old adage of more data isn't always, the old adage that more data is better doesn't always hold in phylogenomics. But this large sample of markers allows us to tease apart our data and look for different kinds of signals. Now, a key issue in phylogenetics that I think many of you are aware of here is this challenge of gene tree heterogeneity. we've known for a long time that as speciation occurs, genes that are being carried around by their constituent populations don't always end up having the same phylogenetic tree. And this can happen for very simple, straightforward reasons, not necessarily involving any kind of gene flow or any kind of hybridization. For purely stochastic reasons, we can end up with situations where the gene tree of a trio of species actually doesn't match the lineage history of the species that are carrying them. So you can see, for example, in this bold line, if you sample these 3 alleles, you end up with a gene tree in which species 2 and 3 are sister relative to species 1. And this is strictly a consequence of very rapid speciation. Basically, genetic drift hasn't had enough time to catch up with the speciation process. And so for many phylogeneticists, it's become increasingly clear that we need to take into account this variation in topology that we might see from gene to gene. And this has led to a variety of new methods, so-called multi-species coalescent methods, that we and others have helped to contribute to. Again, the problem is one of, we have data from many different genes. Do we concatenate them all into a single large supergene and analyze it that way? Or do we allow each gene to achieve its own topology and then summarize the data afterwards? These are 2 very different approaches. There's a lot of discussion in the literature. It's worth pointing out, in fact, that Bruce Ranala's work, I think Bruce is here, really set the stage for many of the coalescent models that my group and other groups have developed. And it's a very exciting time, I would say, in phylogenetics. If I can just digress for a moment, there's a lot of really heated debates out there as to the phylogenetic relationships of important lineages that I think rest on this issue of whether to concatenate or whether to use a coalescent approach. One example is highlighted by my Harvard colleague Chuck Davis, who's a plant phylogeneticist. And you can see here we've got 2 hypotheses for the relationships of Amberella. For botanists, you will know that as the putative sister group to all the other flowering plants, a small diminutive plant that grows in New Caledonia. The prevailing paradigm has been an Amberella alone situation, wherein Amberella is the sister group to all other flowering plants. But a result that Chuck has come up by analyzing transcriptome data is, in fact, that Amberella is actually sister to water lilies, and that pair is sister to all the other flowering groups. Now, again, you may not lose sleep over this, but trust me, people turn blue in the face arguing which one's right. Not the least of which because, NSF, pumped out $7 million at the time to sequence the Amberella genome, partly because of its exalted phylogenetic status. Now, if you look at this question, Chuck did a series of simulations showing that depending on what kind of tree you have and how long those branches are, coalescent methods tend to perform quite well. This is a proportion of correct trees recovered. And concatenation, in fact, will do quite well when the branches in the tree are long, okay? But if branches in the tree are very short, you quickly come into a situation where concatenation, which you see here in red, fails. And so it's distinguishing between these 2 situations, which I think is, it's an exciting time and perhaps a very frustrating time for many phylogeneticists. Now, just to remind you again, these conserved non-coding elements, we'll be talking a lot about these. These are essentially putative enhancers. We can find them in genes by looking for regions of the non-coding DNA that evolve slower than a putatively neutral rate. So we somehow have a tree with branches that expresses the neutral rate around the genome, and we're looking for regions that are evolving slower than that neutral rate but are not encoded within exons. So we call them conserved non-exonic elements. And I think you'll see that in this next slide you can see that these CNEEs actually perform quite well as phylogenetic markers. Here's a comparison of just sort of alignment quality, if you will, for these CNEEs as well as these ultra-conserved elements and introns. And you can see that the number of gaps that we typically get in an alignment is much, much lower for these CNEEs than for the ultra-conserved elements, where, of course, we're using the flanking regions in addition to that core conserved region. And then if we look at a variety of statistics like just the number of undetermined bases, the amount that's trimmed by various alignment trimming algorithms, etc., CNEEs tend to evolve somewhat more slowly than these other markers, but they tend to deliver very robust phylogenetic results. And then a second methodological issue I just want to mention is the algorithm that we'll use to make the tree. This is an algorithm developed by Liang Liu, who is a postdoc in my lab and now is a professor of statistics at the University of Georgia. Basically, Liang's approach involves looking at triplets of taxa. So you have taxa A, B, and C embedded in a larger phylogeny, and you're essentially counting the number of rooted triplets corresponding to any one topology. So you can imagine that in a situation like this, where all of the gene trees for that trio of taxa A, B, and C are the same, it's no surprise that the estimated species tree should look like that particular triplet. But when you get some heterogeneity and you get some minority gene trees, you might still end up estimating the same species tree, albeit with very short branch lengths. And then finally, you have a situation where you have essentially an equal distribution of gene trees. This, of course, would suggest a polytomy in which essentially you've got three species springing from a common ancestor simultaneously. And I want you to remember this situation because in the Palaeognathous example, we're very, very close to a situation like this with some of the nodes. Here's the tree we ended up with. You can see the number of markers and base pairs. Now, intriguingly, these branches are so short, at least in terms of these units we call coalescent units, that it's hard to appreciate that there's actually quite a lot of evolutionary change going on in them. What you can see, though, however, is that the rheas fall as sister group to this larger group containing kiwis and emus and cassowaries. And if you recall, this corresponded to the phylogeny produced by Alan Baker's recent studies and contradicts the phylogenies not only from mitochondrial DNA from Alan Cooper's lab, but also from a limited nuclear data set that was recently published in Current Biology by a Japanese group. The rhea is clearly placed incorrectly in these trees, we think probably because they were using concatenation and not taking into account gene tree heterogeneity. So again, very short branches, but highly significant. Again, if we concatenate the data, whereas with the coalescent methods, we see a really remarkable level of consistency despite those short branches. When we concatenate the data, we tend to get flip-flopping of clades as is expected when there's a high level of gene tree heterogeneity. Now let's just look how short some of those branches are. What we see here is, again, these triplets that describe different relationships of the rhea. So here's rheas with emu, cassowary, and kiwi. That's the dominant pattern. Here's rheas with a different relationship to other groups. And you can see that for all three of the markers, those gene tree triplets are almost equally distributed. That means it's very, very tough to tell which is the true topology, and that we're very, very close to a region called the anomaly zone, wherein if you look at more than three lineages, the most common gene tree is actually different from the species tree. And this is a situation where concatenation is guaranteed to fail. We have some predictions from coalescent theory. I won't go through the details, but we can sort of estimate how short those branches are in sort of coalescent units given this distribution of gene trees. And suffice it to say that it's close to, but not entirely a polytomy. Very, very ancient rapid radiation. Here you can see a situation where that sort of explains this anomaly zone situation where the most common gene tree, here we have essentially a distribution of gene trees across our data set, with the most common gene tree being the one farthest on the left. And you can see that for none of the three markers is the most common gene tree the same as the estimated species tree. This is again a situation where coalescent methods work well, but concatenation is likely to fail. You can see here, for example, that the species tree estimated is actually the fourth most common gene tree here, the fifth most common gene tree for introns, and a surprisingly low frequency gene tree for the UCEs. So all this sort of analysis of DNA sequences is all well and good, but in many ways is kind of unsatisfying because we don't see any hard evidence for major genomic changes that can corroborate phylogeny. So we also looked at transposable elements. Transposable elements, of course, have been used in phylogenetics for a number of years and are very useful in terms of providing a rooted description of your tree. They tend to insert in the genome in different locations, and one can use those insertions to essentially as molecular synapomorphies to create clades. And we've done this for a variety of transposable elements, including these chicken repeat ones. This is what they look like in alignments. You can see there's clear evidence of a transposable element insertion here, and we can infer that that insertion occurred, once this subset of species diverged from their ancestor. And so here's a tree that we get from transposable elements. It's actually identical to the coalescent tree with rheas as sister to this Australo-New Zealand clade. So we're very confident about this tree, and we're moving forward with our genomic analysis. So let's now talk about how we're going to use this tree to understand the genomic basis of flightlessness. We're essentially going to do a series of tests that scan the genome for changes that have occurred in parallel on these branches in red. These are the flightless branches, again distinguished from those of the tinamous, which can fly, and of course those of all the other birds, the so-called neognates, that comprise the sister group. So we're going to look at patterns of protein evolution, patterns of evolution of these conserved regulatory elements to ask ourselves which genomic compartment seems to be evolving more consistently and in parallel on these flightless lineages relative to the other compartments. So the first thing we looked at was patterns of amino acid convergence. And those of you who sort of keep up with this literature, you'll know that there have been some very interesting, albeit controversial, examples of sort of proteome-wide bases for adaptations. Things like adoption of invasion of marine environments, evolution of echolocation, some of them fraught with statistical difficulties. In our case, what we've done is we've laid out for every protein in the genome, essentially, the number of, I guess I should say for every pairwise comparison, the number of substitutions that we see that are divergent between that pair and when you're comparing the amino acid sequences, and those that are genuinely convergent. And the reason why it's important to scale the data this way is because as we get more divergent changes, we expect a lot of convergence to occur just by chance. And so what we're really looking for are a cluster of points above this line suggesting a higher than expected number of convergent amino acid substitutions. We don't see that in our data. We don't see any striking evidence for a high level of convergent amino acid substitution. This isn't to say that convergent substitution in some individual genes might be important, but from a proteome-wide perspective, we don't get a large signal. Let's look now not at convergent amino acid change, but adaptive amino acid change. And I'm sure many of you are familiar with algorithms such as PAML or HiFi that allow you to look at patterns of adaptive evolution, unusually high rates of amino acid substitution on these flightless branches. And in fact, we find a moderate number of genes in the low hundreds, depending on your false discovery rate, that appear to be evolving adaptively on these convergent flightless lineages. These are an interesting set of genes. They contain many developmental regulators. Here you just see sort of a Go analysis of some of those adaptively evolving genes. And so these are exciting candidates to follow up with in terms of molecular evolution. Let's now look briefly at the, I want to again remind you of these CNEs and to remind you that they're involved with gene regulation. They're essentially enhancers. Let's now look at how we're going to look at parallel evolution in these non-coding markers. Essentially what we're going to do is we're going to look at the pattern of branch lengths on the so-called neutral tree. Again, these branch lengths are derived from a putatively neutral set of sites around the genome. We're going to scan the genome for regions that are highly conserved. And as you can see from this CNEE, there's virtually no evolution along this, throughout the entire history of birds. But there's a subset of these elements which are evolving faster on the flightless lineages, which you can see here in red, than in the remaining volant lineages. And it's the signature of accelerated evolution or perhaps relaxation that we're particularly interested in. Here's another view of that same data wherein we have a non-coding element, about 250 base pairs, which shows a high degree of conservation, not only among the neognaths, the sister group to the Palaeognathous, but also in the tinamous, and shows some evidence for a higher number of substitutions in those flightless lineages. This is evidence that that element is becoming relaxed and presumably has lost its function. We've scanned the genome for evidence of these sorts of bouts of acceleration. When we look at a single flightless lineage and ask how many elements become relaxed on that lineage, we find a fairly large number, okay? And we can then ask how close is the nearest gene that it might be regulating, et cetera. But by exploiting convergence, we can narrow down this rather large universe of elements. When we consider two lineages, we actually cut it more than in half with about 1,270 elements. Those elements that show acceleration on 3 different flightless lineages are only about 250 of them around the genome. If we look at 4 lineages, that number drops to 66. And we even have, when we include the MOA in these examples, that number drops to about 21 elements around the genome, all of which are accelerating on the flightless lineages, solely on the flightless lineages. So this is a really interesting subset. We've done a bunch of permutation tests to ask, are these numbers significant? Should we write home about these patterns? And so we've basically done a series of tests using hi-fi and other methods that compares trios of flightless species and asks about the extent of acceleration in those trios versus trios in a different trio of birds, in this case, birds that exhibit vocal learning. We chose this because unlike loss of flight, vocal learning is the origin of a new trait, and we think there might be some different genomic dynamics for that sort of trait than for loss of flight. And then finally, we look at elements that, how many elements are accelerating on a random trio of lineages. So these are sort of our controls. Now what's remarkable is that if we look at, this is sort of a rather dry statistical portrayal of our results, but here we've got p-values on the x-axis. This is looking for evidence of rate accelerations on either vocal learners, which you see here in red. I can't have my glasses on, let's see. Vocal learners in red, ratites in blue, and random trios in black. And in fact, these are actually, this is looking at the protein data first. What's remarkable is that we see very little evidence of convergent protein acceleration in the ratites. That's similar to data I just showed you. Whereas with the vocal learners, we find a highly significant excess of genes which show convergent acceleration. These are the passerine birds, hummingbirds, which also show vocal learning, and parrots, okay? So we see a really interesting contrast here. The vocal learners seem to have a large contribution of protein evolution to their trait, whereas flightless birds, consistent with what I showed you earlier, shows very little evidence of a contribution of protein evolution. Let's look now at the number of CNEs showing convergent relaxation in these two traits. Again, for the ratites, we see a much higher than expected number of convergent accelerations, whether it's 2 lineages or 3 lineages. Notice for the vocal learners, we see very little evidence of a non-coding contribution to this trait. Very few instances where the CNEs are accelerating on lineages leading to vocal learning. So we think this might be a very interesting contrast between loss of flight on the one hand and the origin of vocal learning on another, with a much higher contribution of regulatory change in the loss of flight situation. I just want to end on a few empirical notes. So for every one of these non-coding elements, we can sort of speculate what gene is it, what gene's expression is being regulated by that element. As a first pass, we can look at the closest gene. It's not always going to be the closest gene, but that's a reasonable first guess. And my graduate student, Phil, is looking at those closest genes and asking where are they expressed in developing chickens and in developing emus. Intriguingly, a lot of the genes with showing relaxation of their associated enhancers seem to be expressed in the limbs, both the forelimbs and the hind limbs. These are two examples of some of our highest scoring genes. So this, I think, is an exciting prospect to sort of see what parts of the phenotype might be driven by these relaxations of these regulatory elements. Phil is also using this new approach called ATAC-seq. So ATAC stands for Assay for Transposase Accessible Chromatin. It's essentially a method to ask where in the genome, at a particular place and time, do we see evidence for open chromatin? And of course, open chromatin means that transcription factors can access that region and can drive gene expression. He's looking at this. We've done some initial experiments in the limb buds of chickens, and we're embarking on a series of experiments looking at as the keel develops in chickens, what regions of the chromosomes are exposed to transcription factors for gene expression. And we found a couple really intriguing results. The first result is that these conserved non-coding elements are highly enriched in regions that show ATAC-seq peaks for early stage limb development, okay? What that means is that these sort of signatures, these proxies for gene regulation that we've been using based on bioinformatic results are giving us a genuine biological signal because their chromatin is open at a particular early stage in limb development. The second really interesting result are to look for instances where the chromatin is doing different things in a flighted species and a flightless species. For that, we've looked at chickens and rheas. Here's an example where the ATAC-seq peak, so the open chromatin, is falling directly under a conserved non-exonic element, okay? Intriguingly, this is in chicken, hind limb and forelimb. In rheas at the same stage, we don't see an ATAC-seq peak. That means that at that particular place and stage, the chromatin is not open. So this, I think, will provide a very powerful window to guide us as to which enhancer sequences are most worthy of functional follow-up. And so you can see that the project is far from complete. I know you're all hoping for a list of genes that gave rise to flightlessness. But I hope I've convinced you of a couple of things. First, that these conserved non-exonic elements are not only useful for building trees, but are very useful signposts, sort of proxies for the regulatory landscape of particularly in invertebrates. They also occur in invertebrates and plants. The second is that we can exploit instances of convergence to hone in and narrow down the putative list of candidates, whether they be genes or non-coding sequences. Finally, I hope I've shown you that traits like vocal learning, such as we saw with that trio of species, might have a genome-wide, a very different molecular basis than something like loss of flight. Loss of flight, we see a preponderance of signals for the action of non-coding elements, whereas in vocal learning, we see the opposite. We see a strong contribution of accelerated protein evolution on lineages leading to vocal learning. So with that, I'll end and thank our team, which consists of a great group of computational biologists, developmental biologists, and bird watchers like myself. So thank you. Yes, how you doing? Great to see you.
I was wondering if you could say a little bit more about what information goes into calling a CNE a CNE.
Right. Good. Yeah. It's a hidden Markov model, and you've got two states, conserved state and a neutral state, and you're kind of scanning the genome looking for chunks of DNA that fit better on a tree with short branches than with longer branches. So it's a statistical approach. It's somewhat sensitive to how many species you have in your alignment, but it's fairly robust. There's tuning parameters. You can sort of influence how long these elements are, but in general, it's a pretty repeatable exercise. Yeah. Yes. Hey, Gino. Hey,
I had two questions. What's the shortest divergence time that you see flightlessness and non-flightlessness? Sorry, flight-volant versus flightless. What sort of time scales are you talking about?
That's a great question. So Gino's question is, what's the time scale of evolution? Should have mentioned that. It's a very old group. The divergence of Palaeognathous from all the other birds is likely on the order of 110 million years. The ancestor of ratites is debated, but it's probably on the order of about 70 million years. And when you compare the tinamous, which are the flying group, and the non-flying Palaeognathous, that divergence, it's all very compressed at the base. And so it's probably on the order of about 50 million years or so.
And then sort of a follow-up question. It's hard for me to picture, so you're talking about convergent, a loss of function, and then a convergence of that. So generally, when I think of loss of function, then there's drift after that. So how do you get a convergent force that's driving that?
I mean, in some ways, convergence might be an inappropriate term. I think Gino's question was about convergence and loss of function. We're basically observing similar evolutionary forces, whether it's removal of a force like constraint or an active force like selection operating on parallel branches. And so, whether you call it convergence or parallelism or whatever, it's sort of the independent acquisition of these different evolutionary forces. So that's the important point. Yeah. Yes.
How important is island living to this flightlessness? Because with the exception of ostriches and tinamous, a lot of these flightless birds live on islands.
Yeah.
And if you look at the tinamous across Central and South America, you see a lot of variation in their performances and abilities to fly, or differences in like the sagittal keels or their keels on their chest for like being dispersed, like flight re-evolving as a means of dispersal in this new area.
So the question is sort of how important is being on an island? Yeah, actually, that's a really interesting question. I mean, I think there's some very interesting work done by Natalie, there's a student working with David Stedman in Florida showing that island birds, even when they can still fly perfectly well, tend to evolve in the direction of flightlessness. So shorter forelimb elements, longer hind limb elements. Now, whether flightlessness in the Palaeognathous was driven entirely by an island situation, that's probably not the case. I mean, of course, you've got ostriches. I mean, Africa's a big island, I suppose, but more importantly, presence or absence of predators, right? And clearly, flightlessness has evolved in some situations in the presence of mammalian predators. And so it's, I think in many cases it was, and certainly in groups that we haven't studied here, like rails and whatnot, flightlessness was driven by lack of mammalian predators. But I think in the Palaeognathous, it's probably different reasons in different groups. Yeah.
So how are you thinking about convergence and incomplete lineage?
Right, good question. So Ben's question was, how are we thinking about convergence and incomplete lineage sorting? Very good question. You've obviously thought about this a lot yourself. For our purposes, we're fixing the tree, and we're sort of looking for signals of convergence on a fixed species tree. Unlikely to be correct for all portions of the genome, as I was just, haranguing everyone about. We suspect the effect is small when trying to identify conserved regions or accelerated regions, but I think it's a legitimate question, and because we know that different gene topologies can lead to different patterns of substitution and stuff. So let's talk about that more offline. That's a really good question. Yep.
Are there any stem group fossils of the Palaeognathous which may be informative for the evolutionary pattern you're seeing?
Good question. I mean, Julia Clark, she's a collaborator. That's sort of her department. She's the bird paleo person on the project. The fossil record for extant groups is fairly shallow. I think there's a Miocene ostrich, very well ascribable to ostriches. Of course, that's long after ostriches diverged from the other groups. Of course, there's this group from North America called the Lithoornithids, which are extinct now, but they have Palaeognathous palate, which is the defining feature of these birds, and remarkably could fly. So it's an extinct volant group. So I think there will be a point where we can use fossils to help calibrate this group, although we've mostly been focusing on the genomics yet. And I think, that's caused other groups to really forcefully argue for that flight was lost independently, and they got to all these continents not through continental drift, but by flying there. Yeah.
I'm just curious about NCEs and across other flightless birds, or towards flightless bird types, like maybe about chickens or turkeys or things like that. And I mean, not necessarily ratites, but do you see kind of similar patterns in relaxation of selection on these NCEs?
Right. No, that's a good question. I mean, it's really remarkable the preponderance of relaxation of CNEs we see on these ratite branches. As I showed you a bit, if you look at sort of a random trio of birds, we don't see that pattern very often. I think your question's kind of getting at, well, might we see a suggestion of relaxation if we have birds that are really crummy flyers like chickens and other galliforms? And it's, I think conceivably. I think this is a very new area. I think if we can exploit these issues of multiple losses or diminutions of flight, I think that's going to be a very powerful approach. We haven't looked in detail at that yet, but I think it's, this whole issue of is flight lost gradually, or how is it specialized for different niches, I think that could show up in the genome as well. So, yeah.
Along those lines, do your analyses yield insights on a sequence of sort of these changes that are part of the overall package?
Right. That's a great question, Andrew. I mean, one of my hopes for this project is that we can actually place a sequence on the loss of or the changes in phenotype based on the genome. And so, loss of keel, loss of forelimb elements, changes in body size. I'm thinking we might be able to place those in some sort of temporal order based on when they're related elements are relaxing. And we're not there yet, but it's definitely on our to-do list. Yeah, it'd be really cool.
mean, my sort of naive thought is they got to be able to run fast before they can lose their wings, or do you have even speculation?
I've never seen a bird lose flight, so you know. It's, but I mean, I know what you're talking about, and I think and it's intriguing. You know, these are really important questions, but and I think be able to use genomic patterns to help figure it out. Yes.
So, my history dates back to someone who was very good at obtaining eggs of many of these species.
Oh.
The Sibley
Let's
era.
talk.
The Sibley era.
Okay.
The illegal Sibley.
Yeah.
And my question is there anything that comparative developmental genetics of formation or lack of formation of the keel can tell us about really this issue of convergence versus parallelism
Right.
of the structure?
Yeah. No, that's. I think that's a very good point. Examining the pattern of morphogenesis of the keel or pattern of loss of digits.
During morphogenesis.
Well, so in terms of genes, virtually nothing is known about the keel. No one's really looked at it. There have been, I'm trying to think, there's a guy in the UK who's looked at TBX5, but again, and I believe that was with the keel in emus, but other than that, there's like nothing known. So some of Phil's work is basically going to ask, well, you know, what genes do we see differentially expressed in a emu sternum versus a chicken sternum, just sort of starting from that. But I think that the forelimbs are much better studied, and there we actually have some candidates that we can look at in more detail and stuff. So, something like that.
Yeah. So in ratites, if I remember correctly from one of your former postdocs, the sex determination is not CW like the rest of the birds.
Well, so sex determination was something I was questioning. It's probably similar. The sex chromosomes are quite different.
Okay.
Yeah, and so many birds have a very differently sized Z and W chromosome. In the ratites, they're virtually the same size. There's only a tiny part that's sort of sex specific. And so Tim is actually quite interested in sex chromosome evolution. So he's been looking at that. And there's also these issues of what's called dosage compensation, whether the heterogametic sex can sort of regulate its gene expression so it matches that in the homogametic sex. And for the ratites, the evidence out there for that is unclear. In mammals, it's very well established that it works, but in birds, it's been equivocal. So I think the ratites may be an interesting place to look at that. So, Marilyn, yes.
So thinking about the keel, which is where the pectoralis muscles insert,
Right.
attach.
Right.
If you don't have a keel, do you have pectoralis, you don't have flight muscles, or do you?
I suspect the flight muscles the development of flight muscles are important for generating the keel. They probably work together.
But if
So-
you don't have a keel, are they something to attach to? I mean, we have breast muscles, we don't fly.
True. No, absolutely. So again, it's sort of terra incognita. I think there's a lot of ablation experiments to do to sort of remove the muscles, see if that changes keel development, that sort of thing. But I mean, those are good questions because I think we don't want to naively assume that the keel sort of evolves in isolation from the muscles. So, yeah.
Scott again.